In this video, we will be discussing how to design good experiments. This figure shows respiration rates of 10 frogs that was measured shortly after the frogs had been captured in the wild and brought into the laboratory. So that's the pre-treatment. Then, right after the respiration rates were measured, an insecticide was added to the aquarium water of all 10 frogs at a dose similar to what frogs might experience in their natural habitat. Then after 4 weeks of exposure to this insecticide treatment, the respiration rate of the same 10 frogs was measured again. Do note that these are the same individual 10 frogs measured in the pre-treatment and in the after 4 weeks treatment that you can see these two categories on the x-axis. We call this a within subjects or paired design because we're measuring something on the same individual in two different points in time. Does it look like insecticide affects respiration rates? You would have to do some statistical test to provide adequate evidence of whether there really is an effect or not. But even without doing a statistical test, we can guess that it doesn't look like there's an effect of insecticide on respiration rate in these 10 frogs. So what do you think? Is this a good experimental design? Has the experimenter controlled for all possible extraneous factors so that only the effect of insecticide is being tested? No. We don't know what other things might be happening in these 4 weeks besides just the insecticide treatment. So if we want to know what the effect is of the insecticide treatment is, we have to also control for any extraneous factors that don't have to do with insecticide treatment. From the results shown in Part A, the upper graph alone, the results suggest that insecticide has no effect on the respiration rate of frogs. However, the results reported just in Part A there were in fact only some of the results obtained in this experiment. Actually, 20 frogs were captured for this study, and the respiration rate of all 20 frogs was measured shortly after the frogs had been captured from the wild and brought into lab. However, the aquarium water for the second group of 10 frogs was not treated with the insecticide. The respiration rate of this control group of 10 frogs was also remeasured after 4 weeks. The complete set of results obtained in this experiment is shown in Part B, the lower graph. Assuming that no mistakes were made in the experiment or in the collection of data, and that all the frogs were in generally good health when captured, what do you think is the biological explanation for the results shown in graph B? The control animals demonstrate that over time in captivity, respiration actually increases. What's a possible explanation for that? Well, it could be that the animals have been affected by the stress of capture and that the new captive environment when they were first brought into laboratory made them stressed out and reduced their respiration rates. But after they got used to living in lab for 4 weeks and they were used to this new environment, then that stress response went away and now their respiration rate is higher, which would actually be probably their normal respiration rate. However, the respiration rate of the insecticide-treated frogs did not change over time. So what's a possible explanation for that? Why didn't theirs also increase? It could be that the insecticide is continuing to cause a stress state that is reducing their respiration rate in some way. Based on all of the data collected in this experiment, it could be concluded that insecticide reduces respiration rates of frogs. And in order to really tell, you really just compare the insecticide treatment group and the non-insecticide treatment group after the 4 weeks, and that really tells you that there is a difference in these two treatments. So the lesson from this study is that it's important to control for time-related variables, and if you're measuring things over time. And you must always compare experimentally treated animals with controls that, with the exception of the independent variable under investigation, have experienced identical conditions throughout the experiment. experiment. So in this case, the control is controlling for any other extraneous variables that might have an effect. And so you can see just the effect of the insecticide, not all the other extraneous factors. You could also think about other ways to reduce variability or to look at effects of handling time or stress or whatever. You could let the frogs, all the frogs, habituate to lab after 4 weeks of no treatment for either of them, and then apply the treatment and see what happens there as well. So there are other ways of trying to look at the effects of time and handling. You could handle some more and some less. You could figure out other ways of stressing them. But the importance is that you have, in this case, you need to have some kind of control, some kind of experimental design that takes out all extraneous factors and only looks at the variable of interest, which in this case is the effect of the insecticide. Here's another example that can show you how different comparisons can be made to answer different questions. So in this experiment, the respiration rates were measured in all animals at 30 degrees Celsius at time 0. These baseline respiration rates were used to separate the animals into 2 groups. One of the groups, the experimental group, was then subjected to a new temperature, 20 degrees Celsius, while the other group, the control group, remained at the original temperature. So they stayed at 30 degrees. So the control group shown in the open white circles remained at the same temperature over the whole 90 minutes, whereas the experimental group started at the warmer temperature of 30 degrees and then were subjected to a new temperature, 20 degrees, and then given 90 minutes to kind of acclimate. So they all were measured at time 0 and at time 90 minutes. The control group stayed at the same temperature. The experimental group in the black circles started out at a warmer temperature and then went to a cooler temperature. And just note that normally there would be error bars added to these values. These represent several individuals, not just one individual, but the person who made this graph just left them out in this particular graph because it was too confusing to have error bars all over the place. What they wanted to demonstrate is the different comparisons. So let's talk about some of the different possible comparisons you could make between, you basically have 4 mean values. You have the control group at time 0, the control group at 90 minutes, the experimental group at time 0, and the experimental group at 90 minutes. So these comparisons, A through D, show some ways you could compare the data. So if you first look at comparison A, that's comparing the experimental group at time 0 to the experimental group at 90 degrees. And what this comparison shows is so that would be a within subjects or a paired kind of test that is going to show you the difference due to temperature plus all the time-dependent extraneous variables such as habituation, learning, prior handling, stress, whatever. So you've got 2 things going on there. So if you compare just comparison A, looking within the same subjects at time 0 and time 90, you've got 2 different things going on there. You have time potentially affecting things as well as temperature. So you can't say what is just due to temperature. So that's not a good comparison to make on its own. If you look at comparison B, that is comparing between subjects. So now you have completely different individuals. You're comparing the control group and the experimental group at just time 90. This is showing you the difference due to temperature alone, because the control group is controlling for any possible effects of time. So comparing the experimental versus control at time 90 just tells you what's the difference due to temperature, and it takes out any other extraneous factors as long as your baseline of both groups is similar. Comparison C is looking again within subjects of just the control group, and this is looking at just the extraneous effects of time and has nothing to do with temperature, right? Because the control group stayed at the same temperature the whole time. So here you can see if you found some kind of significant difference between those two groups, that would suggest that there are effects that aren't due to temperature that are also going on. So that's just due to the time-dependent extraneous variables alone and not due to temperature. Comparison D is comparing between subjects at time 0, and that can be a good comparison to make just to see if there's any kind of baseline difference between your two groups. Now, if you do a good job of assigning your subjects to the two groups where there's no bias, then for any reason, whether it was due to size, like all the big frogs were in one group and all the small frogs were in the other, or sex, all the males were in one group, all the females were in the other, or whatever, as long as you have good representation of the variability within the frogs in both groups, then their baseline data should not be different. And in that case, if their baseline data are not different, if comparing D, where you're comparing between groups at time 0, if there's no significant difference there, you can really just compare B, which is between the different groups at time 90. And that will help you answer the question you're actually interested in, is what is the effect of temperature on respiration rates in these animals? So there are a lot of things you need to consider when designing a good experiment. So for example, you need to think about what are your treatments? What are you interested in understanding or knowing or learning? For the experiments in this class, with our limited time and resources, and because most of you are newer to designing experiments, it's best to keep it as simple as possible. It's best to limit to 2 main treatments. Like that last experiment, the main treatment that you're interested in is temperature. You end up having to look at things over time as well, but that's not the variable of interest. The variable of interest is temperature, and they just stuck to 2 temperatures, 30 degrees and 20 degrees, or whatever. You just have 2 temperatures. So keep it as simple as possible. Then you also want to think about how are you going to assign the subjects to treatments. It's very important to have it as balanced as possible so that both treatments have a good representative, unbiased sample. Usually you can randomly assign subjects to groups, and if you have a large enough sample size, then it usually ends up, if you have a large sample size and you randomly assign them, then you usually have good balanced design where both groups represent the same population at the beginning, or to start with the baseline. But occasionally you may end up having some kind of unbalance within your baseline or your first treatment or your pre-treatment or whatever. So, or your two groups in general aren't well balanced, and that can cause havoc in interpreting your data. So it is best to make sure that you do have some kind of balance. So if you have, let's say you had 20 people and you didn't have 10 males and 10 females, you had 18 males and 2 females, well, first of all, you'd want more females than that. But you might split them into males and females and then randomly assign the males to the 2 treatments and randomly assign the females to 2 treatments. But the idea is that you sometimes have to not only randomly assort, it just depends on the situation, but in general, you want to take any possible sources of bias out of how you get your subjects into treatments. You also need to think about any effects of treatment order. So as we discussed, we saw that there are some times when there are extraneous things that can happen over time, and so you need to control for that. So there's different ways you might possibly affect you control for treatment order. Why does the order usually matter? Because variables of like time of day, learning, fatigue, handling, stress, et cetera, can all have effects. So you need to think about how you can deal with that. And I will show you a couple different options for dealing with that, with treatment order. You need to think about what specific extraneous factors might have an effect that you should try to control for. So some examples in the kinds of human physiology experiments we're doing in this class, some things that you want to try to control for if you can include things like stress, fatigue, time of last meal, time and amount of last caffeine, amount of exercise, kind of exercise. Did they run up the stairs right before doing the experiment? Maybe you should tell them to take the elevator, walk slowly, give them time to catch their breath before you start an experiment. How much sleep they had in general or the night before, whether they are adjusted to the measurement protocol, maybe give a practice round just so they know what's coming, so that the order of treatments doesn't matter as much, because sometimes if you do it the first time, you do the measurement, they're kind of like, I don't know what's going on, and they get a little kind of anxious or something, and then that may affect things like heart rate. So you might have to give them some time to adjust to the measurement protocol. In this class, sex shouldn't be a factor since we should have mostly biological human female subjects in this class. But it's possible that we could have people who don't fit all the same category, and that may have some effects. Size could affect, whether we have like larger people versus smaller people, how much exercise they normally get, temperature in the room. One day it might be warmer, the next day it might be cooler, temperature of the subject. So someone might just be cold. One subject might be cold, another subject might be really hot because they, just run hotter or whatever. Extraneous noise in the room could have an effect. Rest between trials, is there a placebo needed? So there's a lot of different extraneous factors. You should think about what things might have an effect on your experiment and what you want to remove, and how can you do that? How can you account for those things? How can you take as much variability out and just look at the variable of interest? Having a larger sample size is usually better. You couldn't do a test on one individual and then say you know anything from that result of just that one individual. Humans are really variable, so larger sample sizes help us reduce the influence of that variation. You need more individuals to get a reliable estimate of what a typical individual in that population really is. So if the data are really variable and you have a small sample size, the study could end up inconclusive. There may be an effect that you can't pick up because you have too much variability and a small sample size. So having a big sample size is generally good. However, getting enough subjects to participate and having enough time to measure a large number of participants is not trivial. It's not a trivial problem. So you want to think about what kind of sample size can you reasonably get. 3 is usually an absolute minimum sample size. You usually can't do statistics unless you have at least 3 subjects or observations. Of course, this does depend on the test, but generally you need at least 3. But that sample size is generally too small, especially for the kinds of tests we are doing where there's a lot of variability between subjects. Conversely, if you try to get a sample size of something like 50 people, that's really just not feasible given the limited amount of time we have to do these experiments, and it would actually be very difficult to get that number of volunteers who could participate. So think of what kind of sample size you can feasibly measure in 2 weeks, and keep in mind that week 1 you may have to spend some extra time making sure you really understand how to use the equipment, and what exactly you're doing and how to do it, and get, it tends to be a little slower at first, and you might even have to throw out a couple data points until you really get it down. So the first week, you may not be able to, just plug through and get a ton of good results. So, think about how much you could reasonably measure in 2 weeks. You can use time outside of lab. You can sign up for times to come in outside of lab time during the week, during regular working hours. But you should also think of other ways you can reduce variability. So again, control for as many extraneous things as possible. Sometimes if you do good within subjects designs, that can reduce variability in ways that you can really look more at the variable of interest. And then you also want to think about, will you make all the measurements within subjects and do pre and post tests on the same individual, like a baseline, and then after they exercise or after they drink caffeine or whatever? Or will you make measurements between subjects so that subjects are totally different in the different treatments? So you have different people in your different treatments. To demonstrate how a within subjects or paired treatment design can be beneficial, this shows the difference between taking into consideration the within subjects versus not taking it into consideration. So here we have measurements taken under 2 treatment conditions, condition A and condition B. And in the left-hand graph, panel A, you can see that it's showing individuals, each pair of points connected by a line represents one subject, the same individual subject who has been measured at treatment A and treatment B. So here is showing, let's say individual subject number 1 at treatment A and subject number 1 at treatment B, and they're connected because it's the same individual. Now, if you did a 2-sample t-test or an independent sample t-test between these 2 conditions, it would just take an average of all the individuals for treatment A, and that's shown here with the error bars. And then it takes an average of all the individuals measured in treatment under conditions B and shows its standard error over here. Now these 2 mean values take no account of which value came from which individual. So it's just taking all the means in A and comparing it to all the means in B, and it's not matching them up in any way. And then it's just comparing them. However, because there's a lot of individual variation, the individual variation is swamping out any possible effect of the treatment. Whereas you can see that if you look at each individual, in general, it looks like most individuals have a higher value under treatment condition B than they do A, even though, like this subject here has a, has a much larger effect than this subject that I drew in originally. But they all have, generally, almost all the individuals have a higher value for B than they do A. So if you take the difference between B and A for each individual, so you're pairing up the values within subjects, then taking that difference and then taking the average of the differences, that's what's shown over here. We'll use a different color. How about blue? So over here in blue is showing it's the average difference. So it's taking the difference for each individual between B and A and then averaging those differences. Now you can see that on average, the difference between the treatments is different from 0. So there is an effect. What this suggests is there is an effect of treatment, and that you have to kind of take into account some of that individual variation by subtracting out the difference within each individual. So this shows you the power of using a paired design or a within subjects design. So in B, where you're using a paired design or within subjects design, there was a significant effect of treatment. But in A, where the within subjects variation is not really taken into account and they just did it independently, there was no significant effective treatment. So doing within subjects or paired treatment can sometimes help when you have a lot of variability. But you do need to keep in mind that you still have to take into account that time could be a possible factor, as we talked about earlier. So this figure shows two possible designs to test the effect of the independent variable when time is possibly a factor. So in design number 1, this is where they're using two groups, and one of the group is a control group that controls for the time-dependent extraneous variables such as previous handling, habituation, learning, developmental age, stress, etc. In this case, you have a control group measured at time 0 and at another time, where you're not doing anything but measuring the effects of time. So if you compared the control group at time 0 versus the control group at time t, the only thing that you're measuring there is the effect of the extraneous variables that happen over time, and you're not measuring the effect of the treatment. Then if you compare the experimental treatment that starts with the same treatment, I guess, as the control treatment, but then is given a different treatment, like a different temperature or whatever, and then measured at another time. When you compare the red individual, the experimental groups at time 0 and time t, that's showing you the effect of temperature and the extraneous variables. So that's two confounding factors. So we already talked about that a little bit. But if you compare between subjects just at time t, then that's just showing the effect of temperature. And of course, again, this depends on your baseline at time 0 being similar between the groups. Another possible way of testing just the effect of the treatment and kind of taking out the order of trials is that you can do this design in design number 2, which is called the treatment order control, or it's also called the crossover design. Uses treatment order to control for time-dependent extraneous variables without designating any one treatment group as the control group. So in this case, you have one group that is measured at one temperature, at a warmer temperature, and then after a certain amount of time measured at a different temperature, a cooler temperature. And then you have another group that you start at the cooler temperature, and then over the same amount of time, after the same amount of time passes, measured at that warmer temperature. So they're crossing over in their the order of the treatments. Then when you compare at time t the two different treatments, what you would do is actually pair. So you take the difference between the two temperatures for each group and then just look at the effect of temperature overall. So that way you have something that starts out as warm and goes to cool, and starts out as cool and goes to warm. And if there's always an effect of temperature, then those means will be different. The differences between those two temperatures will be different. You can also look at the between the two different treatments within a temperature, so at the different times, to see if there's an effect of order. So you would do that at both temperatures and see if there's an effect of order. So this is, in a way, this crossover design, or design 2, is a little more complex, but in both cases, both designs, you can account for possible effects of time or order and just figure out what the effect of the independent variable of interest is. So, so this helps you kind of take out any effects of time or order of treatments. Another thing you want to consider, as I said, is that if your baseline treatment is not the same for your two groups, let's say you have athletes versus non-athletes, and you want to look at the effect of exercise, you might have the athletes measured at time 0 and the non-athletes measured at time 0, and then have them exercise, and then, after they exercise, post-treatment, you measure them both. In that case, you might want to do something where you just take the difference between pre and post-exercise for each group, or you can do percentage change for each group, and then you can see just is the difference different or not. So there are other ways of kind of accounting for the actual treatment. It just depends on what design you're actually doing. So while you're designing your experiments, hopefully you can take into consideration some of the things we've discussed in this video to help you design a really good experiment. Talk it over with your group, think about what you're most interested in and how you can design a really good experiment. It's not as easy as it sounds. It's actually, there's a lot of variation. A lot of times things get published sometimes that have poor designs. And it's really important to learn good experimental design and to practice it over and over again to try and get better at it, because it's a skill that takes time and experience to learn.